In this video, we will discuss about the drug delivery on different combinations of the biomarkers from their designed AND gate logic system. So first they check it for the single biomarkers, that is EGFR on A431 cells, and what they are able to find out when they used both the DNA splitters together, only then they are able to see the cytotoxicity. Also, when they don't use this MMAE, or they do not see any cytotoxicity, and when they use the different biomarkers, then also they don't see significant cytotoxicity, indicating the specificity of their drug delivery system. Thereafter, they move back to the dual marker, that is PDL1, and we will first discuss the panel B. Dual marker, that is PTK7 and EGFR, and in this case they tested on different cell lines, that is A431 cell lines, MDa-MB-468, SCT116, and MCF7. And in all the four cell lines, this A431 cell lines have the high expressions of this PTK7 and EGFR. So they see the significant cell death in this when both the biomarkers are both the biomarkers DNA splitters are used, as in this case H1M1 and H2M3. Then only they are able to see the cell cytotoxicity, whereas this MDa-M8-468 also shows cytotoxicity. However, the cell death is low, and this is relative to the lower expressions of this PTK7 in this MDa-MB-468, whereas other cell lines do not show significant cytotoxicities. So this all further indicates that the specificities of the drug delivery system is to the point that it does not cause cell death when they are not able to find out the biomarkers. Afterwards, they checked it for the different biomarkers combinations also, that is PD1, PDL1, and EGFR in the same four cell lines, and they are able to see cell death for the case of this A431 cell lines when both the biomarkers are expressed. Whereas since these biomarkers are both the biomarkers are not present in the other cell lines, they do not see significant cell death in other cases. Once validating their dual markers individually on the individual cell lines, let me rephrase this sentence. Once validating their dual biomarkers on the individual cell lines, now they wanted to check it in the more biologically relevant system, where they use the mixed populations, that is A431 and Hela-GFP. So these combinations that they have used, and then use the biomarkers, that is EGFR and PTK7. So this Hela does not have both the biomarkers, so it should not undergo any cell death. So when they use both the biomarkers, they are able to see a decrease in the cell death for this A431 cell lines, whereas in the other conditions where they don't use both the biomarkers, or they use the control conditions, that does not have the cytotoxic drug, or they don't have both the ligands, in that case they are not able to see cell death. Their cell proliferation is high, whereas when they use both the biomarkers, and that time they are able to see the cell death. So this further substantiate their point that this drug delivery system can be used with different combinations and with high specificity and minimum side effects. So this is what they finally conclude, this is what they finally come up from the results that are presented in this figure. So let me just go through these third conditions. Here the no ligand conditions means that they have this DNA splitter, but they don't have this ligand, so it will not attach to the cell surface. So internalizations of this drug this cytotoxic drug, M1 and M3, will not occur. It will only occur when these hybridization chain reactions occurs. But since they don't have any ligand, so there will be no SCR, that is hybridization chain reactions. So the cytotoxic drug will not enter into the cells. And the immunofluorescence image also shows the same results that in the case of when both the drugs biomarkers and the cytotoxic drug is present, the A431 cell lines goes down, as can be seen from the panel F and also from this immunofluorescence image. But for the other conditions, we can see that the red signals which corresponds to the A431 cell lines. So this this is some more explanations about the final experiment. So if you need to understand more about this experiment in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.